A team of scientists took old blind mice, reset something inside their eye cells, and the mice could see again. Aging reversed. In a living animal, confirmed. But when they pushed further, something unexpected happened inside the brain tissue, something they still cannot fully explain. We are going to go through every layer of this, from the countdown clock inside each of your cells, to zombie cells, to young blood, to the moment reprogramming overshoots and cells forget who they are. If you want to understand where your biology ends and the future begins, stay with us. Like and subscribe if this kind of deep science is what you come here for. We begin now. You have a clock inside you, not a metaphor. A physical, molecular clock built into every single cell in your body, ticking down with every division, every repair, every time your biology has to copy itself. And most people have no idea it's running. Here's what's actually happening. Your body is made of roughly 37 trillion cells. Those cells don't live forever. They divide, they age, they die, and new ones replace them. That process of replacement is what keeps you alive. Your skin renews itself, your blood replenishes, your gut lining replaces its entire surface every few days. Division is life. But division has a cost. Every time a cell copies itself, it has to duplicate its DNA. All 6 feet of it, coiled and packed into a space smaller than a grain of sand, has to be unzipped, read, and rewritten perfectly. And at the very tips of your chromosomes, at the end of each strand of DNA, sit small protective structures called telomeres. Think of them like the plastic caps at the ends of a shoelace. Their job is to keep the chromosome from fraying, from unraveling at the edges during copying. Every time the cell divides, those caps get a little shorter. A young person's telomeres are long, healthy, protective. But by the time you're in your 40s and 50s, years of division have worn them down. And when telomeres get critically short, the cell gets a signal. Stop dividing. You've run out of road. Some cells respond by dying. That's actually fine. The body clears them and moves on. But some cells do something stranger. They stop dividing but refuse to die. They just sit there, locked in place, secreting inflammatory signals into the tissue around them. Scientists call these senescent cells. Most people, once they hear what these cells actually do to surrounding tissue, start calling them something else entirely. We'll get to that. The shortening of telomeres is confirmed science. Decades of research, thousands of studies. This is established biology. But what's fascinating, and what opens the door to everything we're going to cover, is that telomere shortening is the clock, not the cause. Aging isn't just telomeres wearing down. Aging is what happens when your body's systems start losing the information they need to do their jobs correctly. Here's a way to picture it. Imagine a library. Every cell in your body is a librarian who holds a complete copy of the instruction manual for the entire human organism. Young librarians have clean, well-organized books, sharp, easy to read. But every time they photocopy a page, tiny errors creep in. Smudges, blurred lines. The words are still there, mostly, but the instructions get harder to read. After enough cycles, the librarian can't quite remember what it's supposed to be doing. That's aging at the molecular level, a slow erosion of biological information. Now, the extraordinary thing is this. In the 1980s, a scientist discovered an enzyme that can do something the rest of biology seemed to have ruled out. It can rebuild those telomere caps. It can reverse the shortening. It can, in a very specific sense, rewind the clock. The enzyme is called telomerase, and your body produces it in certain cells, under certain conditions. The fact that it exists at all changes everything about how we understand aging's rules. Because if there's an enzyme that rebuilds what aging destroys, the question isn't whether reversal is biologically possible. The question is why your body ever decided to shut it off. Telomerase shouldn't exist. From an evolutionary standpoint, aging seems like it should be permanent, fixed. Your cells wear down, you die. The next generation takes over. That's the standard script. So when a young researcher named Elizabeth Blackburn and her colleagues discovered telomerase in the 1980s, the scientific community had to sit with an uncomfortable idea. The body already has the tool to slow cellular aging. and it mostly chooses not to use it. Telomerase is an enzyme, which means it's a molecular machine built from proteins and a small strand of RNA. Its job is specific. It finds the shortened ends of chromosomes, uses its built-in RNA as a template, and adds new telomere material back onto the tip. It rebuilds the cap. It extends the runway. Cells that have active telomerase can divide far beyond the limits that other cells face. You already have cells in your body that use telomerase heavily. Your stem cells use it. Your immune cells ramp it up during an infection. The cells lining your reproductive system rely on it. These are cells that need to divide rapidly and repeatedly without burning out, so the body gives them access to the enzyme. Every other cell keeps telomerase mostly switched off. The reason biologists believe this trade-off exists comes down to a danger hiding inside the very tool that grants extra divisions. Telomerase, when unregulated, removes the natural brake on cell division, and a cell that divides without limits, that never gets the signal to stop, is something every person already knows by another name. Cancer. Roughly 90% of confirmed cancer cells reactivate telomerase on their own. They hijack the enzyme, switch it back on, and use it to become effectively immortal within the body. So the system your body evolved isn't careless. Keeping telomerase suppressed in most cells is a controlled sacrifice. You age, yes, but you dramatically reduce the odds of rogue cells taking over. That trade-off made sense for a species where the priority was surviving long enough to reproduce, not living for 150 years. Evolution doesn't plan for longevity beyond reproductive age. Once you've passed your genes on, from evolution's perspective, your continued survival is optional. This is confirmed biology. The telomerase suppression is deliberate, shaped by millions of years of pressure. But here's where it gets genuinely strange. In the early 2000s, scientists began asking whether it was possible to turn telomerase back on selectively, carefully, in the right cells, at the right time, for the right duration. They ran the first experiments in mice, activating telomerase in aged animals with critically short telomeres. The results were striking. Tissues that had been deteriorating began recovering. Organs that had shrunk with age began gaining function. The animals looked and behaved measurably younger. One team described watching the spleen, the brain, the intestines of aged mice regain characteristics they hadn't seen since the animals were young. Confirmed improvements in actual tissue. And then the researchers stopped the telomerase activation and checked for cancer. The mice didn't develop it, at least not in those early short-term trials. The activation had been careful enough, brief enough, that the danger stayed contained. That result opened a door that has never fully closed. Telomere rebuilding works in animals. The brake can be released, briefly without immediate catastrophe. What no one had yet figured out was how to do that in a human cell, where the stakes are incomparably higher. But a different scientist, working on a completely different question, was about to stumble onto something far more powerful than telomerase alone. In 2006, a Japanese scientist named Shinya Yamanaka published a paper that restructured the foundations of biology. His question going in was deceptively simple. If every cell in the body contains the same DNA, the same complete instruction set for building an entire human being, why does a skin cell behave like a skin cell, and a heart cell behave like a heart cell? What decides which instructions get read and which ones stay locked away? The answer is a system of molecular switches. Proteins, called transcription factors, sit on the DNA and control which genes are active at any given moment. A skin cell has certain switches flipped on, others flipped off. A neuron has a completely different configuration. The DNA is identical. The configuration of switches is what makes them different. Yamanaka's team took adult mouse skin cells, fully specialized, fully committed to their identity, and introduced just 4 specific transcription factors, 4 proteins. They labeled them by their initials: OCT4, SOX2, KLF4, and CMY. In the field, scientists call them the Yamanaka factors. What happened to those skin cells was something biology had assumed was impossible. They reversed. They shed their specialized identity. They wound back through developmental time and became what scientists call induced pluripotent stem cells, cells that had forgotten they were ever skin cells, cells that could, in principle, become any cell type in the body all over again. Yamanaka won the Nobel Prize in Physiology or Medicine in 2012. The discovery earned it. But embedded inside that extraordinary result was a warning that the field would spend the next decade wrestling with. When those four factors ran long enough, when the reprogramming went too far, the cells didn't just become young, they became chaotic. They lost all their specialized function. A cell that forgets it's a skin cell also forgets every skill it spent years developing, every protein it knew how to produce, every signal it knew how to send. And chaos in a cell, inside a living animal, looks a lot like a tumor. Full reprogramming, it turned out, was dangerous precisely because it worked completely. It was the biological equivalent of formatting a hard drive to fix a few corrupted files. Yes, the corruption is gone. So is everything else. The field then split into two camps. One group pursued full reprogramming for specific controlled applications, growing replacement tissues and organs outside the body. The other group asked a different question. What if you ran the factors for just a short time? What if you gave cells a partial reset, enough to restore their function, but stopped before they lost their identity? That idea, partial reprogramming, became one of the most contested and exciting areas in all of biology. Early animal results suggested it could work. Older cells, exposed briefly to the Yamanaka factors, showed measurable improvements in function. Their gene activity patterns shifted toward younger profiles. Their mitochondria, the energy-producing structures inside cells, behaved with more efficiency. But the line between enough and too much was razor-thin, and no one yet knew exactly where that line sat in a living human body. That uncertainty didn't stop the experiments. It accelerated them, because a different set of scientists had just started injecting something far simpler into old animals, and the results looked almost too good to be true. Before we go further, there's a number you need to understand, a fixed boundary that sits underneath all of aging biology like bedrock. In 1961, a cell biologist named Leonard Hayflick did something straightforward. He took normal human cells and kept them alive in a dish, feeding them nutrients, keeping conditions perfect, and simply watched what happened when they divided. The expectation at the time was that healthy cells, given ideal conditions, would just keep dividing indefinitely. They didn't. Human cells divided roughly 50 times, give or take, depending on cell type. And then they stopped. Completely. Permanently. No matter how perfect the conditions, no matter how much nutrition, no matter what signals were sent, after roughly 50 divisions, the cells entered a state of permanent arrest. They were alive but finished. This ceiling became known as the Hayflick limit, and it forced a complete rethinking of what aging actually is. The body doesn't wear out purely from external damage. Aging is partly programmed into the cells themselves. The limit is intrinsic, structural, and the mechanism behind it loops directly back to what we've already covered. Each division shortens the telomeres a little more. When the telomeres reach a critical minimum length, the cell's internal monitoring systems detect the damage and trigger the arrest. The countdown clock built into the chromosome tips is precisely what enforces the Hayflick limit. The two discoveries, made decades apart by different scientists, turned out to be describing the same phenomenon from different angles. Here's where it gets more complicated and more interesting. The Hayflick limit varies across cell types in ways that directly shape how we experience aging. Nerve cells in the brain divide rarely after birth, so they aren't running through divisions quickly. Heart muscle cells are similar. These cells age differently from skin or gut cells, which are dividing constantly. The organs that age fastest, that lose function earliest, tend to be the ones whose cells are cycling the most. And there's a detail about the limit that most people miss. The 50 division count isn't reset between generations. A cell that has already divided 20 times and then divides again to produce two daughter cells doesn't give those daughters a fresh count of 50. They inherit the remaining 30. The clock is cumulative, passed from parent cell to child cell, generation after generation. This means the cellular debt accumulates across a lifetime. By the time you're in your 60s, Many of your tissues are running on cells that have only a handful of divisions left in them. The reserve is nearly spent. Now telomerase can extend that reserve. The Yamanaka factors can in theory reset it. But the Hayflick limit revealed something that neither intervention fully addresses. The limit exists for a reason. Cells with unlimited division capacity become dangerous. The ceiling is protective as much as it is restrictive. So when scientists began designing experiments to push past that ceiling, they weren't just solving an engineering problem. They were making a biological argument that the body's built-in ceiling, which evolution set for very specific reasons, can be safely overridden with enough precision. Some of the early animal data suggested they were right. Some of the later data suggested they had underestimated how many other things that ceiling was quietly holding in place. The strangest part of all this is that the most dramatic results didn't come from manipulating the ceiling directly. They came from a completely different angle, from looking at what happens in blood. In the mid-1800s, a French physiologist named Paul Bert stitched two living rats together. He connected their circulatory systems so that their blood would flow between them, shared. The experiment was called parabiosis, and it was largely forgotten for over a century. Then, in the early 2000s, a team at Stanford University brought the idea back. They paired young mice with old mice, connecting their bloodstreams the same way. And what happened to the old mice startled everyone in the lab. The aged animals showed measurable physical improvements. Their muscles recovered faster, their livers regenerated more effectively, their brains showed increased activity in regions associated with learning. The old mice were getting younger, measurably. Biologically, the scientific community's first response was cautious excitement. The results suggested that something circulating in young blood, some signal or combination of signals, was actively holding aging in check, and that as animals age, those signals fade from their bloodstream, and the tissues respond by deteriorating. Several candidate molecules were identified. One of the most discussed was a protein called GDF11, growth differentiation factor 11. Early studies suggested it declined with age and that reintroducing it could restore muscle and brain function in older animals. Papers were published. Science magazines ran with it. The story spread. Then the replication attempts began, and the picture cracked. Other labs trying to reproduce the GDF11 results got inconsistent data. Some found benefits at certain doses, nothing at others. Some found the protein actually increased with age in humans, the opposite of what the early work suggested. The initial findings held up in some tissues, but fell apart in others. The scientific consensus on GDF11 specifically shifted from confirmed to contested, and it remains there today. This is important to understand because it's how science actually works. The parabiosis result, the broad finding that young blood circulation benefits old tissue, held up across multiple labs. That general effect appears real, but the specific mechanism behind it remained elusive. The field had seen the effect without fully grasping the cause. What the controversy did was sharpen the research. If GDF11 wasn't the whole story, then something else in young blood, or multiple things together, was responsible. The scientists began cataloging the differences between young and old blood with increasing precision. Thousands of proteins, hormones, and molecular signals analyzed and compared across age groups. One consistent finding emerged. Old blood contains elevated levels of signals that promote inflammation, cellular stress, and tissue deterioration. Young blood contains higher levels of signals that promote repair, growth, and cellular maintenance. The difference isn't one molecule, it's a shift in the entire biochemical environment that cells live in. Think of it this way. A cell is like a worker whose output depends not just on its own energy, but on the office environment around it. Old blood creates a toxic office. Young blood creates a supportive one. Swap the environment, and the same worker performs differently. Human clinical trials began testing diluted young plasma in older patients. Some showed cognitive improvements in Alzheimer's patients, significant enough to generate serious attention. But the trials were small, early, and far from conclusive. The blood story was alive. The mechanism was murky. And while researchers chased it, a completely different discovery was quietly building into something far more actionable. Scientists had found the cells responsible for poisoning the environment, and they had figured out how to kill them. Picture a cell that has hit its division limit, received the signal to stop, and decided to partially comply. It stops dividing. That part works. But instead of dying and clearing out the way a well-functioning cell would, it stays. It persists. It parks itself in your tissue indefinitely and begins releasing a cocktail of inflammatory proteins into everything around it. Scientists named this secretory state, and what comes out of these arrested cells is called the senescence-associated secretory phenotype. The name is unwieldy, so researchers in the field mostly just say these cells are secreting inflammatory signals. The popular term for them, which stuck because it's accurate, is zombie cells. A single zombie cell can corrupt the healthy cells around it. The inflammatory signals they release don't stay local. They drift through the tissue, changing the behavior of neighboring cells, recruiting immune cells that then cause additional damage, and gradually degrading the function of entire organs. In young, healthy people, the immune system clears senescent cells relatively efficiently. They appear, they get flagged, and immune cells remove them before the damage accumulates. The problem arrives with age, when the immune system's clearing efficiency drops, and zombie cell populations begin to build up. By the time a person is in their 60s or 70s, senescent cells have accumulated in measurable concentrations across the lungs, the kidneys, fat tissue, and the joints. The link between zombie cell accumulation and the specific physical experiences of aging is confirmed across multiple studies. Chronic low-grade inflammation, the kind that underlies cardiovascular disease, joint deterioration, cognitive decline, and metabolic dysfunction, tracks closely with senescent cell load. These cells aren't just a side effect of aging, they are an active driver of it. Here's what made researchers take this seriously as a target. In 2011, a team at the Mayo Clinic ran an experiment in mice genetically engineered so their senescent cells could be cleared on command by administering a drug. The researchers let the mice age naturally while clearing their zombie cells periodically throughout life. The treated mice lived significantly longer. Their tissues stayed healthier, their physical function remained higher. The results were clean and striking enough to validate the approach as a genuine direction. That led to the next question. Could you do this without genetic engineering? Could an ordinary drug find and kill senescent cells in a normal living organism? The answer turned out to be yes. A class of drugs called senolytics, derived partly from existing compounds including a plant flavonoid called quercetinib, showed the ability to selectively eliminate senescent cells in animal studies. The selectivity matters enormously because you can't simply kill every cell that isn't dividing. You destroy neurons, heart muscle cells, and a host of other vital cells that stop dividing for healthy reasons. Senolytics have to find the ones that are actively causing damage. In animals, senolytic treatment extended healthy lifespan, improved organ function, reduced inflammation markers, cleared the toxic environment that was dragging surrounding cells into deterioration. Human trials began. Early results in patients with specific conditions, including a lung disease associated with severe cellular aging, showed promising reductions in senescent cell markers. The field is moving fast. The biology is real. The drugs exist. And while zombie cells were being hunted, a completely different team had built a clock so precise it could tell you not how old your birth certificate says you are, but how old your cells actually are. Your cells know how old they are, and they write it down. Every cell in your body carries not just your DNA sequence, but a layer of chemical tags sitting on top of it. These tags don't change the letters of your genetic code. They change which letters get read. A tag on a gene can silence it completely. Another type of tag can amplify it. The entire system of these tags and how they regulate gene activity is called epigenetics, which means literally above the genetics. What a biostatistician named Steve Horvath discovered in 2013 is that the pattern of these tags across specific sites in the genome changes with age in a remarkably consistent, measurable way. Across different people, different cell types, even different tissues, the same set of chemical markers shifted in the same direction at predictable rates as the organism aged. He built a mathematical model that could look at those markers and output a number, a biological age. Not a guess, a measurement with striking accuracy often within a few years of a person's chronological age, and in many cases, revealing that two people of identical birth years could have biological ages years apart from each other. This is confirmed science. The Horvath clock and its subsequent refinements have been validated across dozens of independent studies. It works. Your epigenome records your age the way a tree records its years in rings, except the record is written in chemistry, at the molecular level, in every cell of your body. The implications hit researchers in two directions simultaneously. First, the clock gave scientists an objective measurement tool they had never had before. Instead of waiting years to see if an aging intervention worked, they could now measure biological age before and after a treatment and get a direct readout. The clock compressed research timelines dramatically. Second, and more unsettling, the clock revealed that biological age and chronological age can diverge significantly based on lifestyle, environment, stress, and disease. People who smoked heavily, who experienced chronic stress, who had certain metabolic conditions, showed epigenetic ages years ahead of their birth certificates. People with unusually healthy habits sometimes showed the reverse. Your biology is keeping score whether you check it or not. Then the obvious question arrived. If the clock runs forward with age, could it be made to run backward? The answer, in at least some contexts, appears to be yes. A small but closely watched human clinical trial called the TRIIM treated a group of healthy older men with a combination of growth hormone, metformin, and a diabetes drug, monitoring their epigenetic clocks throughout. By the end of the trial, participants showed measurable reversal of their biological age on the Horvath clock. An average of about 2.5 years of biological age reversed in roughly 1 year of treatment. The trial was small. The interpretation is still debated. But the fact that the clock moved backward at all, in living humans, under a real clinical intervention, changed what the field considered possible. And if biological age could be measured and reversed at the level of chemical tags on DNA, that raised a question even more specific. Could you reverse aging in a single organ? In a single tissue? Could you target just the eyes, for instance, just the optic nerve, and restore something that had been lost? A team decided to find out. The experiment started with a specific, measurable problem. As mice age, the neurons in their retinas deteriorate. The cells responsible for converting light into electrical signals, the ones that make vision possible, lose function gradually and permanently. By the time a mouse is old, its visual acuity has dropped significantly. The connections between the eye and the brain have weakened. The signal gets through, but faintly. Scientists at Harvard, led by a team working with David Sinclair, asked whether partial reprogramming could restore those connections. They used a modified virus to deliver 3 of the Yamanaka factors directly into the retinal cells of aged mice. Not all 4 factors. 3, carefully chosen to trigger partial reprogramming without pushing cells all the way back to a stem cell state. The results, published in 2020, were confirmed and reproducible across multiple rounds of the experiment. The treated mice showed measurable improvement in visual function. Their retinal cells recovered the gene expression patterns of younger cells. The electrical signals traveling from eye to brain strengthened. The mice could see better. More striking still, the team then created a model of glaucoma, a condition where the optic nerve is damaged under pressure, similar to what happens in human glaucoma. They applied the same reprogramming approach to mice with active nerve damage. The nerves regrew, the connections re-established, function returned to tissue that had been actively injured. This was the first confirmed demonstration of partial reprogramming, reversing aging, and repairing damage in a specific sensory system inside a living adult mammal. The scientific community's response was measured, but serious. The result was real. The eye is also, in terms of complexity, a relatively accessible target. It's self-contained. The blood-brain barrier, which protects the brain from most molecules circulating in the body, doesn't fully apply in the same way to the retina. Delivering a virus to the eye is far simpler than delivering one to, say, the cerebral cortex. But the implications pointed outward. If the approach worked in the eye, the logical next target was the nervous system more broadly. Could reprogramming restore aging neurons? Could it be used to treat conditions like Parkinson's disease or the general cognitive decline that comes with aging? The team and others began running those experiments, and the results were promising, with a caveat so significant it reframed the entire conversation. When partial reprogramming was applied to neurons in the brain, some cognitive function did improve in aged mice. Memory performance on standard maze tests got better. Certain markers of neural health recovered. But something else happened in the brain tissue that the researchers hadn't fully anticipated. A small subset of cells appeared to overrespond to the reprogramming signal. They began expressing gene patterns that didn't match any normal adult cell type. The cells hadn't become cancerous. They hadn't died. They had become something in between, something the researchers couldn't fully classify. The effect was minor in scale. The animals remained healthy. But the finding introduced a warning the field couldn't ignore. The brain is the one organ where identity, memory, and personality live. Resetting aging in the brain means, in some molecular sense, resetting the record of experience those cells have accumulated. The question of what exactly gets erased and whether function can be restored without touching anything deeper is now one of the most urgent and least resolved questions in the entire field. And it sits directly underneath something even harder: the question of what you actually are, if your cells are reset to a younger state. The brain is where aging reversal gets complicated, in a way that goes beyond biology. Every other organ can be thought of as a machine with a function. The liver filters, the heart pumps, the kidneys clean the blood. When those machines age and deteriorate, restoring their cellular youth seems straightforwardly good. A younger functioning liver means better metabolism. A younger functioning heart means lower cardiovascular risk. There's no philosophical complication. You get a better performing organ. The brain is different. Your neurons are not just cells doing a job. They are the physical record of everything you have ever learned, every relationship you formed, every memory you carry. The connections between neurons, the specific pattern of which cells talk to which other cells, and how strongly encode your personality, your skills, your sense of who you are. Neuroscientists call this pattern the connectome. And it's the closest thing to a biological definition of identity that science currently has. Here is the tension. A neuron that has been active for 60 years has accumulated 60 years of molecular changes. The chemical tags on its DNA, the strength of its synaptic connections, the proteins it produces in response to experience, all of it has been shaped by a lifetime. That's partly aging, but it's also partly the neuron becoming more specifically, more precisely you. When partial reprogramming begins resetting a neuron's epigenetic state, it's rolling back the clock on both things simultaneously. The deterioration and the experience sit in the same molecular layer. The Harvard eye experiment suggested this could be done carefully enough, that function improved without catastrophic loss. The neurons regained their ability to transmit signals efficiently. But the retinal cells in the eye don't store memories. They transmit visual data. The stakes of resetting them are comparatively low. In the brain's memory regions, the hippocampus and the cortex, the situation is far more fraught. Animal experiments applying reprogramming factors to aging brain tissue have shown improved neurological performance in some respects. Older mice navigated mazes more effectively. Their neural circuits fired with more youthful efficiency. But a close examination of what happened at the cellular level revealed something the headline results didn't capture. Certain memory-associated patterns in the neurons shifted during the reprogramming window. The changes were subtle. The animals didn't appear to forget their training, but the molecular signatures of specific learned behaviors showed some evidence of disruption. This is early data in mice, and interpreting behavioral changes in rodents involves genuine uncertainty. Scientists are careful not to overclaim, but the theoretical concern is real and is taken seriously by researchers in the field. There's a thought experiment that helps make this concrete. Imagine your brain as a notebook. The paper itself, the physical substrate, ages and degrades over time. Partial reprogramming could refresh the paper, make it hold ink better, let the cells communicate with more clarity. But the ink is also real. The writing is also real. And the two are not fully separable. Any intervention that touches the paper also risks smudging the ink. The field's current working assumption is that careful, targeted, limited-duration reprogramming can thread this needle. That you can restore enough cellular health to extend cognitive function without erasing the patterns that make a person who they are. The assumption is reasonable. The data supporting it, in humans, is nearly non-existent. And while researchers work toward that data, a completely different angle on brain aging had already produced results that were easier to interpret. Results that came from looking at the body's second brain, not the one in the skull. There are more microbial cells in your gut than there are human cells in your entire body. That statement has been refined and debated over the years. But the current scientific consensus puts the ratio at roughly 1:1. And in terms of genetic diversity, the microbes in your gut carry about 150 times more unique genes than your entire human genome. The ecosystem living inside your digestive system is not a passenger. It is an active participant in your biology, and it ages with you, or rather, it degrades with you. In young, healthy people, the gut microbiome is diverse. Hundreds of different bacterial species, each playing specific roles in digesting food, producing vitamins, regulating the immune system, and sending chemical signals that influence brain chemistry through the gut-brain axis, a direct communication pathway between intestinal neurons and the brain. A rich, diverse microbiome is one of the clearest biological markers of good health. As people age, that diversity collapses. Certain species decline. Others, including bacteria associated with inflammation, become disproportionately dominant. The microbiome shifts from a balanced ecosystem to a less stable, more inflammatory one. And because the gut communicates constantly with the immune system, a deteriorating microbiome actively accelerates the immune deterioration that drives systemic aging. The causal arrow runs in both directions. Aging degrades the microbiome. A degraded microbiome accelerates aging. What happens when you reverse that degradation? A series of animal experiments answered this with unusual clarity. Researchers transplanted gut microbiomes from young mice into old mice, and then watched what happened across multiple organ systems. The old mice showed measurable improvements in immune function. Their gut lining, which had thinned and become more permeable with age, began recovering structural integrity. Markers of systemic inflammation dropped. And in one study published in a major biology journal, old fish who received microbiome transplants from young fish not only showed improved health markers, but lived significantly longer than untreated animals. The young microbiome extended life. That is a confirmed result in animals. Human data is more limited and more complicated. Fecal transplant procedures exist and are used clinically for specific gut conditions. Researchers have begun studying their effects on aging markers in older patients. Early results suggest improvements in inflammatory markers and immune function, but the trials are small and the long-term effects remain unknown. What's clear is that the gut microbiome is a legitimate lever in the aging system, one that operates somewhat independently of the cellular aging mechanisms we've covered so far. You can have cells with excellent telomeres and a well-functioning epigenetic clock and still be biologically older than you should be, because your gut ecosystem is in disarray. This also means that lifestyle factors affecting the microbiome, diet, sleep, antibiotic use, physical activity, are not just loosely associated with aging. They are mechanically connected to it through a pathway that is now well understood. But even the gut microbiome sits downstream of something more fundamental. Inside every one of your cells, including every cell in your gut lining and your neurons and your muscles, there are small structures that produce essentially all of the energy your body runs on, and they are failing, slowly, measurably, with direct consequences for every system we've discussed so far. A mitochondrion is about the size of a bacterium. That's deliberate, because about 2 billion years ago, something that was a bacterium got absorbed into a larger cell, and instead of being digested, it struck a deal. It would provide energy. The host cell would provide protection and resources. That partnership became the foundation of all complex life on Earth, including you. Every cell in your body contains hundreds to thousands of mitochondria, depending on how energy-intensive that cell's function is. A resting skin cell might have a few hundred. A heart muscle cell, which never stops working, carries several thousand. Their job is to take the nutrients from food you eat and convert them into a molecule called adenosine triphosphate, the universal energy currency that powers virtually every chemical reaction in your body. When mitochondria work well, your cells are energized, responsive, and capable of repair. When mitochondria deteriorate, the consequences ripple outward through every system, and they deteriorate with age faster than almost any other cellular structure. Here is why. Mitochondria generate energy through a process that involves passing electrons along a chain of protein structures. That process is efficient, but not perfectly clean. Some electrons escape the chain and react with oxygen to produce what are called reactive oxygen species, essentially molecular sparks. These sparks damage nearby structures, including the mitochondria's own DNA. Mitochondria have their own separate genome, distinct from the DNA in the cell's nucleus. It's smaller, less protected, and far more vulnerable to damage. As mitochondrial DNA accumulates errors over a lifetime, the energy production process becomes less efficient. The mitochondria produce less adenosine triphosphate. They also produce more reactive oxygen species, which causes more damage, which reduces efficiency further. It's a self-reinforcing cycle, and it begins in measurable ways as early as your 30s. The cellular consequences extend beyond energy. Poorly functioning mitochondria send distress signals that activate inflammation. They trigger cellular stress responses that push cells towards senescence, the zombie state we discussed earlier. Mitochondrial dysfunction appears consistently as an upstream driver of the aging cascade, not just a byproduct of it. What do you do about it? Several approaches are under active investigation. One involves a molecule called NAD+, nicotinamide adenine dinucleotide, a compound that mitochondria rely on heavily for energy metabolism. NAD+ levels decline significantly with age. Supplementing with its precursors, compounds like nicotinamide mononucleotide or nicotinamide riboside, has been shown to restore mitochondrial function in aged animals, with measurable improvements in energy output, muscle endurance, and metabolic health. Human trials are underway. Early data shows that NAD+ precursor supplementation does raise NAD+ levels in older adults and appears safe. Whether those elevated levels translate into the dramatic functional improvements seen in animals is still being studied. A separate approach involves exercise, specifically the type of exercise that forces mitochondria to work hard and then recover. High-intensity interval training triggers a cellular response called mitophagy, where damaged mitochondria are broken down and replaced with new, healthier ones. The cells essentially prune their worst-performing power plants and rebuild from their better ones. This is a confirmed mechanism. Mitophagy, induced by exercise, measurably improves mitochondrial quality in older adults. The mitochondrial story reveals something important about aging interventions in general. Some of the most powerful tools available aren't exotic or experimental. They work through pathways that have existed in your biology for your entire life. But the tools being developed in labs right now are something else entirely. And one of them has already been tested in humans, producing results that left scientists genuinely uncertain about what they were seeing. There's a molecule inside every one of your cells that functions like a master key. NAD+, nicotinamide adenine dinucleotide, is involved in over 500 chemical reactions in the human body. It helps mitochondria produce energy. It activates enzymes called sirtuins that regulate DNA repair, inflammation. It supports the machinery that detects and fixes broken strands of DNA. Without adequate NAD+, essentially every major maintenance system in the cell functions at reduced capacity. In a young body, NAD+ levels are high. The cellular environment hums with repair activity. Damage gets caught quickly. Sirtuins stay active. Mitochondria run efficiently. Then, across a lifetime, NAD+ levels fall. By the time a person reaches their 60s, NAD+ concentrations in most tissues have dropped to roughly half of what they were in youth. Half the key supply. Half the access to the systems those keys unlock. The connection between NAD+ decline and aging is well established in animal models. Studies in mice have shown that restoring NAD+ levels to youthful concentrations produces striking effects. Aged muscle tissue regains strength. and endurance. Liver function improves. The cardiovascular system shows markers of better health. In some studies, lifespan extended significantly. The mechanism isn't magic, it's maintenance. When NAD+ is restored, the sirtuins come back online. DNA repair improves. Mitochondrial efficiency increases. The cascade of senescence and inflammation that drives biological aging slows measurably. Now, you can't just swallow NAD+ directly. The molecule doesn't survive digestion and transit into cells efficiently enough to raise tissue levels. What researchers found is that giving the precursors, the raw materials the body uses to make NAD+, works far better. Two precursors in particular, nicotinamide mononucleotide and nicotinamide riboside, raise NAD+ levels reliably in humans when taken orally. Both are commercially available as supplements. Both have been studied in human clinical trials for safety and efficacy. The safety data looks clean. People tolerate these compounds well across the doses studied so far. The efficacy data is more nuanced. Human trials have confirmed that these precursors raise NAD+ levels in the blood. Some trials have shown improvements in specific markers, including muscle function in older adults and metabolic measurements in people with metabolic conditions. But the dramatic whole-body rejuvenation seen in mouse studies hasn't fully translated into human outcomes yet. Whether that gap exists because the human trials have been too short, too small, or because humans are genuinely different from mice in this regard, is an active debate. The field's honest position is that the mechanism is real, the target is valid, and the full human picture remains incomplete. What's notable is that NAD+ sits at the intersection of almost every other aging mechanism we've discussed. It connects to mitochondrial health, to epigenetic regulation, to DNA repair, to inflammation. Any aging intervention that improves NAD+ function is effectively touching multiple systems simultaneously. That convergence is exactly what made some researchers confident that a more comprehensive intervention, one that combined NAD+ support with hormonal balance and metabolic control, could produce measurable age reversal in actual human beings. A small clinical trial put that idea to the test, and the results moved the epigenetic clock in a direction that surprised even the scientists running the experiment. In 2019, a clinical trial called the TRIIM trial published its results. The name stands for Thymus regeneration, immunorestoration, and insulin mitigation. The combination of goals tells you something about the approach. The researchers weren't targeting one aging mechanism. They were targeting several simultaneously. The trial enrolled 9 healthy men between the ages of 51 and 65. Small group, intentionally narrow. This was a pilot study designed to test safety and generate initial data, not to prove anything definitively. The participants received a combination of human growth hormone to stimulate immune system regeneration, metformin to manage the metabolic side effects of growth hormone, and androgen that declines significantly with age. Over the course of 1 year, the team took repeated blood samples and ran epigenetic clock analysis on the participants' cells. The clocks moved backward. On average, participants showed roughly 2.5 years of biological age reversal on 2 different epigenetic clock measures. And crucially, 6 months after the trial ended, the reversal appeared to be holding. The participants' biological ages hadn't snapped back to their pre-trial levels. This was the first time a controlled human intervention had produced confirmed, measurable reversal of biological age using a validated molecular clock. The result generated serious scientific attention. It also generated appropriate caution. 9 participants is a very small sample. The trial had no placebo control group, meaning there was no group of similar men taking fake treatments for comparison. Men experienced significant hormonal changes between 50 and 65, and some natural fluctuation in epigenetic clock measurements is possible. The researchers acknowledged all of this openly, but the signal was strong enough that the TRIIM trial became a foundational reference point. It demonstrated that the epigenetic clock can be moved in humans. It demonstrated that a combination approach targeting the immune system, metabolism, and hormone balance could produce measurable biological age change in 1 year. A larger follow-up trial, TRIIM-X, enrolled more participants across a wider age range, and added women to the study group. Its results were not yet fully published at the time this research was compiled, but preliminary data continued to suggest epigenetic age reversal in treated participants. The TRIUMPH trial also highlighted something the broader field hadn't fully confronted. The thymus gland, a small organ sitting behind the breastbone, was not just a target of the intervention. It was a central character in the aging story. In young people, the thymus is active and full-sized, producing the immune cells that defend the body against infection and cancer. By the time most people reach middle age, the thymus has largely shrunk and been replaced by fat tissue. Its output of immune cells has dropped dramatically. And as immune surveillance weakens, the body becomes less capable of clearing senescent cells, less capable of fighting cancer, and less capable of resisting the cascade of inflammation that drives aging across every system. The TRIUMPH trial's growth hormone component appeared to partially regenerate thymic tissue in some participants. Immune markers improved. The implication was extraordinary and largely underreported. One organ's regeneration might be a lever that amplifies aging reversal across the whole system. That idea sent researchers back to an animal with a very different relationship to aging, one that seems to have solved a problem that has destroyed every other organism that's faced it. The naked mole rat is, by any reasonable standard, an improbable creature. It lives underground in the dry savannas of East Africa, in colonies of up to 300 individuals. It's hairless, wrinkled, and roughly the size of a sausage. Its teeth sit outside its lips. It cannot regulate its own body temperature. It spends its entire existence in dark tunnels, eating roots and tubers, moving in organized columns through a social hierarchy built around a single breeding queen. And it essentially does not age. A mouse of equivalent size lives for roughly 2 to 3 years. The naked mole rat lives for 30 years or more. Individual animals have been documented surviving past 37 years in captivity, which is unheard of for any rodent. More striking than the longevity is how these animals reach it. They don't show the gradual physical decline that characterizes aging in other mammals. Their cancer rates are extraordinarily low. Their cardiovascular function stays consistent well into old age. They maintain reproductive capacity far longer than comparable species. Females remain fertile into their 20s. By every biological measure, naked mole rats compress almost all of their physical decline into the final few months before death, rather than spreading deterioration across years the way humans do. Researchers studying these animals have identified several mechanisms that appear responsible. One confirmed finding is that naked mole rat cells produce an unusually pure form of a large sugar molecule called hyaluronan, which accumulates around their cells and appears to create a physical barrier that blocks the early stages of tumor formation. When scientists removed this barrier in lab experiments, mole rat cells became susceptible to cancer in ways they normally aren't. A second finding involves how mole rat proteins are synthesized. Their ribosomes, the molecular machines that build proteins inside cells, produce proteins with fewer errors than those of most other mammals. Higher protein quality means fewer misfolded proteins accumulating in cells, and misfolded proteins are a significant driver of cellular stress and aging. A third mechanism involves their response to oxidative stress. Mole rats appear to maintain higher levels of antioxidant activity in their tissues, managing the molecular spark damage from mitochondria more effectively than other rodents. None of these mechanisms is unique to mole rats in isolation. Humans have hyaluronan. Humans have antioxidant systems. Humans have quality control mechanisms in protein synthesis. The extraordinary thing about naked mole rats is that all of these systems operate simultaneously at unusually high efficiency, creating a compounded resistance to the multiple processes that cause aging. This means aging isn't a single problem with a single solution. The mole rat demonstrates that longevity requires tuning multiple systems at once, which is precisely the conclusion that every other avenue of research is converging on independently. There's one more animal whose biology challenges the foundations of what we think aging even means. An organism that under the right conditions appears to reverse its entire developmental process and begin growing young again from scratch. What it reveals about the theoretical limits of aging reversal is both fascinating and, depending on your perspective, deeply unsettling. There is a jellyfish, roughly the size of a fingernail, that can do something no other multicellular organism on Earth has been confirmed to do repeatedly and reliably. When it is stressed, starving, injured, or simply old, it reverts. Its adult body transforms backward through its developmental stages, returning to a juvenile polyp state, and then grows into a new adult again. The creature is called Turritopsis thornii. Scientists first observed the reversal in a lab in the 1980s. For years, it was treated as a curiosity, a biological footnote. Then researchers began studying the mechanism behind it. What happens during the reversal is a form of transdifferentiation. Cells in the adult jellyfish change their identity, converting from one specialized cell type to a completely different one without passing through a stem cell stage in between. A muscle cell becomes a nerve cell. A skin cell becomes a digestive cell. The entire organism restructures itself from the inside out. This is extraordinary because it suggests that the boundary between cell types, the wall that keeps a liver cell and a heart cell, is not as permanent as mammalian biology makes it appear. Under the right conditions, with the right molecular signals, cellular identity can be fundamentally rewritten. The jellyfish isn't immortal in any absolute sense. It can be killed by predators, infection, or environmental conditions. Biologists describe it as potentially biologically immortal under ideal conditions, meaning it has no known intrinsic upper limit on lifespan from aging alone. Studying the genome of Turritopsis thornii revealed something the scientific community found remarkable. The genes associated with DNA repair, DNA replication, and cell identity regulation show significant expansions compared to related jellyfish species that age normally. The jellyfish carries more copies of these genes and shows higher activity in the relevant pathways. Humans carry some of the same genes. The implication is that the capacity for this kind of radical cellular renewal is partly written into animal biology at a deep level. It hasn't been lost. It's been suppressed or reduced in organisms like us, likely because the control mechanisms that make complex mammalian life possible require strict cell identity stability. But the jellyfish demonstrates that suppressed is not the same as impossible. Alongside the jellyfish, a freshwater organism called Hydra presents a different but related case. Hydra are tiny tentacled animals that reproduce primarily by budding off copies of themselves. Their cells divide continuously and appear to maintain stem cell populations so efficiently that they show no measurable increase in mortality rate with age. Studied Hydra populations in labs have been kept alive for years, with no observable aging effect. They don't become more likely to die as time passes. They simply live until something external kills them. The mechanism here is different from the jellyfish reversal. Hydra maintain constant stem cell renewal throughout their bodies. Their cells never accumulate enough division history to trigger the senescence pathway. They continuously replace aging cells before the deterioration can compound. Both organisms sit far from human biology on the evolutionary tree. Translating their mechanisms into treatments for mammals is a problem of staggering complexity. But they do something important for the scientific conversation. They prove that the aging process, across the full sweep of animal life, is a set of parameters that evolution has set differently in different lineages, and parameters that can in principle be adjusted. The question that follows is both practical and profound. Even if every mechanism of aging could be perfectly addressed, is there a ceiling above which no biology can survive? Is there a point where the accumulated weight of damage simply cannot be undone? In 1997, a French woman named Jean Calment died at the age of 122 years and 164 days. She holds the confirmed record for the longest documented human lifespan. She rode a bicycle until she was 100. She smoked until she was 117. She attributed her longevity to olive oil and chocolate, which delighted the press and frustrated the scientists trying to understand her. No one has matched her record since. And a study published in Nature in 2016 argued that this wasn't a coincidence. The researchers analyzed maximum reported lifespans across countries over decades and found that improvements in survival rates had effectively plateaued at the extreme upper end, around 115 years. They proposed that 115 years represented a natural ceiling for the human lifespan, set by the accumulated biological damage that no known mechanism can fully overcome. The paper generated fierce debate. Other researchers argued the analysis was flawed, that the sample sizes at extreme ages are simply too small to draw reliable statistical conclusions, and that if aging interventions advance significantly, the apparent ceiling will move. The debate has not been resolved. What does the biology say? Several processes suggest genuine hard limits exist, at least without radical interventions. One is the simple accumulation of somatic mutations, errors in the DNA of ordinary body cells that build up over a lifetime. By extremely old age, many cells carry hundreds of mutations. Some tissues show evidence of dozens of competing mutant cell populations, each with slightly altered behavior. The body manages this through immune surveillance, but the surveillance itself degrades with age. Another hard limit candidate involves the cross-linking of proteins. Over decades, certain proteins in the body form abnormal bonds with each other, and with sugars through a process called glycation. These cross-links accumulate in tissues like the heart, the arteries, and the skin, making them stiffer and less functional. Some of these cross-links are extremely difficult to reverse. Certain drug candidates are in development to break them, but the challenge is significant. A third process is the gradual loss of protein quality control throughout the cell. Old cells accumulate damaged and misfolded proteins that the cellular cleanup systems can't clear fast enough. The resulting clutter interferes with normal cellular function in ways that compound over time. Each of these processes is in principle addressable. None has been addressed comprehensively in a living human being. David Sinclair, among others, has argued that the concept of a hard lifespan ceiling is based on observing biology as it currently operates, without interventions, and that extending it by decades or more is achievable with sufficient scientific progress. Others, including biologists focused on the fundamental physics of cellular damage, argue that entropy sets a genuine ceiling that biology cannot fully escape. The honest answer is that no one knows where the true ceiling is. Gene Calment reached 1202 without any aging intervention science has yet developed. With the tools being built now, the question of whether 150 or 180 or beyond is possible remains genuinely open. What is known is that even discussing that ceiling changes something important. Because a world where large numbers of people live dramatically longer isn't just a biological question. It is a social, ethical, and political transformation with consequences that no current institution is equipped to handle. Every major civilization in human history has been built around a fundamental assumption. People are born, they grow, they work, they age and they die. Generation replaces generation. Knowledge transfers, power shifts, culture evolves. The rhythm of human civilization is partly the rhythm of human mortality. Remove aging from that equation, and essentially every social structure humans have built needs to be reconsidered from the foundation. Start with the simplest economic consequence. Retirement systems in every developed country are built on the assumption that people work for roughly 40 years and then draw on pension and social support systems for another 15 to 20. Those systems are already under strain with current demographic trends. In a world where the average person lives to 150 or beyond, the math of every pension fund, every social security system, every healthcare financing model collapses entirely. Researchers studying the economics of radical life extension describe it as a civilizational accounting problem with no obvious solution within current frameworks. Either the working years must expand dramatically, the retirement model must be abandoned, or wealth distribution systems must be restructured from scratch. Then consider medicine. The entire field of oncology, cardiology, and geriatric medicine is organized around the diseases of aging. If aging itself becomes treatable, the disease burden shifts. Infectious diseases, accidents, and mental health conditions become proportionally dominant. The training pipelines for doctors, the research funding priorities, the pharmaceutical industry's entire portfolio, all of it would need to reorganize around a fundamentally different distribution of human illness. Political consequences are equally significant. Democratic systems assume that power changes hands across generations, that new cohorts with different experiences and priorities regularly enter the electorate and leadership positions. A society where people in power remain biologically 40 for a century presents challenges to political renewal that political theory has barely begun to examine. There are equity concerns that researchers in bioethics are already raising urgently. Aging reversal technologies, at least in their early forms, will be expensive. Access will follow existing economic inequality. A world where wealthy people stop aging while poor people continue to age on the old timeline isn't just unfair. It creates a biological stratification of society that has no historical precedent. The environmental dimension is rarely discussed but significant. Earth's resources, its food production, its water systems, its carbon absorption capacity are calibrated to a human population that turns over across generations. A world population that largely doesn't die of aging, grows until something else limits it, and the planetary systems sustaining life face corresponding strain. None of this means aging reversal is wrong. It means that the scientific achievement and the social adaptation required to absorb it are both enormous, and that the science is, in some respects, moving faster than the frameworks designed to evaluate its consequences. The regulatory structures governing clinical trials were built to evaluate drugs that treat specific diseases. Aging reversal doesn't fit neatly into that framework, because aging itself isn't classified as a disease in most regulatory systems. Scientists applying for approval to test aging interventions in healthy humans face bureaucratic categories that don't anticipate what they're doing. A coalition of researchers has been pushing for aging to be formally reclassified as a disease, which would unlock clearer regulatory pathways and more straightforward funding mechanisms. The debate is ongoing. The regulatory landscape is shifting, but slowly, while the science accelerates. And underneath all of the social and regulatory complexity, the original scientific mystery still sits unresolved. The unexpected finding in the brain tissue, the cells that partially reprogrammed and became something that couldn't be classified. That anomaly hasn't gone away, and the deeper researchers look at it, the more it seems to point towards something fundamental about biological identity that the field hasn't fully reckoned with yet. Your immune system does two jobs most people know about. It fights infection. It hunts cancer. But it does a third job that almost no one thinks about. And that third job may be the most important one for understanding why aging accelerates the way it does. Your immune system clears debris. Every day, across every tissue in your body, cells are dying, becoming senescent, producing misfolded proteins, and generating molecular waste. The immune system's cleanup crews patrol constantly, identifying this debris and removing it before it accumulates into something damaging. When the cleanup runs efficiently, tissues stay healthy, inflammation stays low, the aging cascade slows. When the cleanup falters, everything else accelerates. The age-related decline of immune function is called immunosenescence, and it is one of the most well-documented processes in all of aging biology. As the immune system ages, several things happen simultaneously. The production of new immune cells slows as the bone marrow becomes less efficient. The diversity of immune cells decreases, leaving the repertoire of threats the immune system can recognize narrowed. Inflammatory signaling increases even in the absence of active infection, a state researchers call inflammaging. Chronic low-grade inflammation driven partly by the immune system's reduced ability to resolve responses it starts. Here is the link that ties everything together. Zombie cells, the senescent cells we discussed earlier, are normally flagged and cleared by immune surveillance. Young immune systems do it poorly. As immune clearance drops, senescent cell populations grow. As senescent cell populations grow, the inflammatory signals they release further damage the immune system. Each system's decline accelerates the others. The same feedback loop applies to mitochondrial debris, misfolded proteins, and cellular waste more broadly. A young immune system stays ahead of it, an old one falls behind. And once you fall behind, the backlog compounds. This makes immune rejuvenation one of the highest leverage targets in the entire aging field. If you can restore immune surveillance to a younger operating level, you simultaneously improve clearance of senescent cells, reduction of inflammation, protection against cancer, and resistance to infection. A single target with cascading benefits across every other system. The TRIIM trial, which we covered earlier, targeted thymic regeneration partly for this reason. The thymus produces T cells, the immune system's adaptive response units, and its decline with age directly reduces the diversity and quantity of immune protection available. Partial thymic regeneration produced measurable immune improvements in the trial participants. Other approaches are targeting the bone marrow directly, attempting to restore the stem cell populations that generate immune cells in the first place. Early animal studies suggest this is feasible. Human trials are in early stages. There's a confirmed and somewhat surprising finding that connects the immune system to the epigenetic clock. Immune cells age epigenetically faster than most other cell types. The biological age registered by immune cells in older adults tends to run ahead of other tissues. This suggests that immune aging is both a driver of systemic aging and a sensitive early indicator of it. Measuring immune cell epigenetic age may become one of the most practical ways to monitor whether an aging intervention is actually working in a real human body. Before visible physical changes appear, before organ function measurably shifts, the immune cells may show the signal first. And one specific organ, one that most people have never thought about after childhood, turned out to be central to immune age in ways that stunned even the researchers studying it. Most people know they have a thymus the way they know they have a spleen. Technically, yes. Functionally, vague. It sits behind your breastbone, above the heart, and in childhood it's a busy, productive organ. In adolescence, it begins shrinking. By the time most people reach their 40s, much of it has been replaced by fat tissue. By 60, it's largely inactive. What the thymus does during its active years is produce T cells. Specifically, it takes immature immune cells from the bone marrow, puts them through a rigorous maturation and selection process, and releases them as fully functional T lymphocytes, capable of recognizing an enormous range of threats. The thymus is the training academy of the immune system, and its output is what gives the body adaptive immunity, the ability to recognize specific pathogens and mount targeted responses. When the thymus shrinks, T cell production drops. The diversity of the immune repertoire narrows. The body becomes less able to respond to novel threats, less able to detect and destroy early-stage cancer cells, and less able to clear the senescence cells that drive systemic inflammation. Every downstream problem we've discussed in the immune system traces back, at least partly, to this one organ going quiet. The TRIIM trial's growth hormone component appeared to partially reverse thymic shrinkage in some participants. Imaging showed increased active thymic tissue. Blood markers confirmed improved T cell output. The immune systems of the participants behaved in certain measurable ways, more like those of younger people. That result prompted a separate, more focused investigation. A team using a different approach, targeting a specific molecular pathway involved in thymic tissue maintenance, ran experiments in aged mice attempting direct thymic regeneration. The results showed that the organ retained the capacity to regrow functional tissue even after decades of decline. The cells hadn't disappeared. The biological program for thymic function was still present, just suppressed. A confirmed human trial of thymic regeneration was completed in older adults using a modified drug approach. Participants showed measurable increases in thymic tissue volume, confirmed by imaging, and improvements in the immune markers associated with T cell diversity. This was genuine organ regeneration in living adult humans, documented and published. The implications reach beyond immunity. The thymus connects to cancer surveillance, to autoimmune regulation, and to the inflammatory environment that shapes aging across every tissue. An older person with a more youthful thymus isn't just more resistant to infection. They carry a fundamentally different biological environment in every organ. What this reveals about aging more broadly is that the system is deeply interconnected. You cannot separate immune aging from cellular aging, from epigenetic aging, from mitochondrial aging. Each one influences all the others. This is why single-target interventions, however impressive in isolation, consistently underperform expectations when applied to the full complexity of a living aging body. The organisms that age most gracefully, the naked mole rat, the hydra, the Turritopsis jellyfish, all maintain multiple systems simultaneously at unusually high efficiency. They don't fix one thing, they maintain the whole. This is the argument that researchers pushing for combination therapies make, and it's the argument that brings us to the strangest confirmed finding in the entire field. An organism that spends decades showing almost no biological aging across any measured system. An animal whose existence forced researchers to reconsider whether aging, for a sufficiently tuned biology, is truly inevitable at all. Actually, we covered the naked mole rat and the jellyfish already. What comes next is the question those organisms made unavoidable. If aging can be compressed, slowed, and potentially reversed, is there a version of this that runs into a wall biology cannot cross? And is that wall about the cells, or about something deeper in the physics of living systems? Every system that processes energy produces disorder. That is a law of physics, not a theory, not a working hypothesis. The second law of thermodynamics states that in any closed system, entropy, meaning disorder, increases over time. Energy does work, and in doing work, it generates waste, heat, and degradation that cannot be fully recovered. A living organism is not a closed system. It takes in food, processes it, expels waste, and uses the energy extracted to maintain order against the thermodynamic current. Life is, in a sense, a continuously powered argument against entropy. As long as you keep feeding energy in, you can maintain the ordered structures that define a living body. But the argument gets harder to sustain over time, and the reason is subtle. Every process that maintains order generates small amounts of irreversible damage. The copying of DNA introduces errors that repair systems fix imperfectly. Mitochondrial energy production generates reactive oxygen species that damage cellular structures. Proteins misfold. Crosslinks accumulate. Immune surveillance misses some targets. Each individual error is tiny. Across decades, the compound interest on all of these tiny failures builds into the measurable deterioration we call aging. The theoretical question the field is now confronting is whether perfect maintenance is physically possible. If you corrected every DNA error the instant it formed, cleared every senescent cell immediately, maintained NAD+ levels and mitochondrial quality continuously, and kept the epigenetic clock from advancing, could you maintain a human body indefinitely? Some researchers argue yes, in principle. They point out that the damage mechanisms are all identifiable and addressable with sufficient precision. Aubrey de Grey, a biogerontologist, has spent decades arguing that aging is an engineering problem with engineering solutions, and that every major class of aging damage has a plausible intervention pathway. Others argue that the accumulation of somatic mutations alone sets a genuine ceiling. By extreme old age, many tissues carry hundreds of mutations per cell across billions of cells. Managing that mutation load while avoiding cancer requires immune surveillance of almost inconceivable precision and coverage. Some biologists believe the required precision exceeds what any biological system can sustain, regardless of how well tuned it is. A third perspective focuses on information theory. Every copy of a complex system introduces noise. DNA repair reduces the noise but cannot eliminate it. After enough generations of copying, the accumulated noise degrades the system's ability to run its own maintenance programs. The aging process is, on this view, a slow loss of biological information that cannot be fully reversed because the original reference copy is itself being degraded. These are theoretical frameworks, and the honest position is that no one has tested them against a biological system sophisticated enough to determine which one is correct. The experiments required to answer this question simply haven't been possible yet. What we can say with confidence is that no natural organism has achieved indefinite survival from aging alone. The jellyfish reversal and the hydra's apparent biological immortality are the closest confirmed examples, and both exist in organisms of radically simpler architecture than a mammal. Whether human biology can be made to approximate those examples through targeted interventions stacked in combination is the question that will define the next century of medicine. But it isn't the most unsettling question the field is sitting with right now. The most unsettling question is one that emerges specifically from brain aging reversal, and it isn't about biology at all. Here is the thought experiment the field has been quietly wrestling with. Imagine a person at 60 years old. Call her Maria. Maria has lived 60 years. She has memories encoded in specific synaptic patterns across her neural tissue. She has personality traits built from decades of experience, trauma, joy, relationships, and learning. Her preferences, her fears, her instincts, the way she responds to kindness and to threat, all of it is written in molecular detail across the specific configuration of connections between roughly 86 billion neurons. Now, suppose a partial reprogramming intervention resets the epigenetic state of Maria's neurons to approximate what they were at 30. The mitochondria in her brain cells regain youthful efficiency. The transcription factor activity shifts toward a younger profile. The cells become biologically 30. Is Maria 30? The surface answer is no. Her memories are encoded in synaptic strength, not in the epigenetic state of individual neurons. Resetting the epigenetic clock in a neuron should not in principle erase the synaptic connections that encode memory. The cell can be biologically younger without its network connections being deleted. But the surface answer doesn't fully hold, and here is why. The epigenetic state of a neuron influences which genes it expresses. The genes it expresses determine which proteins it produces. The proteins it produces shape how the synapse functions. The synapse's function is part of what encodes the memory. These layers are not cleanly separable. Resetting the epigenetic state of a neuron changes its molecular behavior. Changing its molecular behavior shifts how its synapses function. Shifting synaptic function modifies the memory trace, potentially subtly, potentially in ways too small to notice, in a behavior test on a mouse, but real at the molecular level nonetheless. The preliminary evidence from brain reprogramming experiments in aged animals suggests that the functional improvements in cognition come with some molecular reorganization in memory-related regions. The mice perform better, but what exactly is happening inside the cells that are performing better remains genuinely unclear. This matters enormously for human application. Cognitive improvement is a goal almost everyone would endorse. But if the mechanism of improvement involves some degree of molecular reorganization in memory and personality circuits, then aging reversal in the brain is simultaneously a therapeutic intervention and something closer to a modification of identity. Bioethicists studying this question have begun arguing that brain aging interventions require an entirely separate regulatory and consent framework from every other medical intervention. When a surgeon operates on your liver, your identity is not at stake. When a molecular intervention reorganizes the epigenetic state of your neurons, even partially and even beneficially, something more fundamental is in play. The people most likely to want brain aging reversal are the people most likely to be experiencing cognitive decline. And cognitive decline itself compromises the capacity to give fully informed consent to an intervention that might alter who you are in subtle ways. This is not a hypothetical concern being raised to slow progress. It is a sincere philosophical and medical problem that the field will have to resolve before brain reprogramming moves from animal experiments to human trials at scale. And it connects to a broader question that the entire aging reversal field is now forcing into view. A question about who decides, who gets access, and what happens to society when the ability to live dramatically longer is real but unevenly distributed. History has never faced this specific problem before, so there are no established frameworks for handling it. But the shape of the problem is already visible, and researchers in medicine, economics, and political science are beginning to describe what it looks like. Every medical technology follows a predictable adoption curve. It begins expensive and rare, available only to the wealthiest and most connected. Over time, if the technology can be manufactured at scale, costs fall and access broadens. Vaccines, antibiotics, statins, and antiviral drugs all followed this arc. Today, they reach billions of people who couldn't have afforded them in early form. The optimistic case for aging reversal follows the same arc. Early treatments are expensive. Wealthy people access them first. Over decades, costs fall, manufacturing scales, regulatory frameworks adapt, and access broadens to the general population. The same way that life-saving cardiac surgery went from experimental and elite to standard and broadly available. The pessimistic case argues that aging reversal is categorically different. Most medical technologies treat acute conditions. A person needs bypass surgery once. A person takes antibiotics for a week. The treatment is finite. Aging reversal, by its nature, is continuous. It requires repeated intervention across decades. The economic model isn't a one-time procedure. It's a subscription to continued biological youth. Maintaining that subscription across a lifetime is a different economic proposition from treating a single disease. And the gap between those who can maintain it and those who cannot would not be a temporary inequality that closes as costs fall. It would be a permanent, compounding biological divergence, one population aging on a natural timeline, another aging on an extended or arrested, one the downstream consequences are profound. A society stratified by biological age rather than chronological age is also stratified by energy, cognitive capacity, and physical ability in ways that map directly onto existing economic inequality. The wealthy would remain physically capable, mentally sharp, and reproductively viable for far longer than the poor. The gap between these groups would widen not just economically, but biologically. Some economists studying this scenario describe it as the most severe equity challenge in human history. Because it operates at the level of the body itself, access to education can be expanded by building schools. Access to biological youth cannot be easily democratized in the same way, without either universal subsidization at enormous cost or regulatory restriction that slows the technology's reach to everyone. The regulatory dimension adds further complexity. In most countries, clinical trials are required to demonstrate safety and efficacy against a specific disease or condition. Aging, in most regulatory frameworks, is categorized as a natural process rather than a disease. This means that interventions targeting aging directly face ambiguous regulatory pathways. A coalition of scientists has been pushing to have aging formally reclassified as a disease within the International Classification of Diseases, the global standard used by regulators and insurers. If aging is a disease, it becomes a regulatory target, insurance becomes potentially applicable, and the development pathway for interventions becomes clearer. The reclassification debate is ongoing. The arguments against it are partly philosophical, that aging is a universal condition rather than a pathology, and partly practical, that reclassifying it would create enormous pressure on insurance systems and public health budgets. Meanwhile, the technology advances, and the gap between what science can do and what society is prepared to govern continues to widen. That gap is widest around the question of what comes after successful aging reversal. Because the full consequence of dramatically extended human lifespans isn't just a social and economic problem, it is a planetary one. Earth carries approximately 8 billion people. That population has grown rapidly over the past 2 centuries, as medicine, agriculture, and sanitation extended human lifespans and reduced infant mortality. The planet's agricultural systems, water supplies, energy infrastructure, and carbon absorption capacity are already under measurable strain. The current global population projection assumes a demographic transition. As countries develop, birth rates fall. As medicine improves, lifespan extends moderately. The model suggests the global population peaks somewhere between 9 and 10 billion people later in this century, then stabilizes or slowly declines. Those models assume people continue to die of aging at roughly current rates, introduce effective aging reversal at scale, and the demographic model breaks. Death rates from age-related causes, which represent the overwhelming majority of all deaths in developed countries, drop sharply. The population doesn't peak and stabilize. It continues rising. The land, water, and energy required to sustain it expand without a natural ceiling. This is a physical and ecological reality, not an ethical argument against developing aging reversal. But it means that any civilization that successfully implements aging reversal must simultaneously solve multiple large-scale coordination problems that humanity has never managed to solve, even under present conditions. Food production would need to expand dramatically while becoming far more efficient, requiring advances in agriculture, vertical farming, cellular meat production, and resource management that are individually challenging and collectively unprecedented. Water systems would face similar pressure. Energy demand would climb at the same time decarbonization is required. Population distribution would shift in ways that further stress resources. Urban density would increase. Migration pressure between regions with different levels of aging reversal access would intensify. Some researchers studying this problem argue that the answer is a sharp decline in birth rates, essentially a civilizational negotiation where longer individual lives are balanced by fewer new lives beginning. Whether such a negotiation is feasible, or politically realistic, or culturally acceptable across the diversity of human societies, is a question that goes well beyond biology. Others argue that the resource and population problem is solvable through technology, that the same pace of innovation that would produce aging reversal would also produce the energy, food, and resource solutions required to support a larger, long-lived population. Space colonization appears in some versions of this argument as a long-term pressure valve. The honest, scientific position is that neither the problem nor the solution is fully characterized. The ecological consequences of successful aging reversal at scale are underexplored relative to the pace of the underlying science. The people working on reversing aging and the people working on planetary resource sustainability are largely separate communities with limited coordination between them. That coordination gap is one of the quieter risks in this entire story. Not a reason to stop, a reason to start thinking harder, and while civilization-scale questions remain open, the science itself has reached a moment of genuine uncertainty at its frontier. The unexpected finding in the brain reprogramming experiments, the cells that became something unclassifiable, is one piece of a broader pattern, a pattern of results that confirm the general direction of the research, while revealing that biology contains surprises the models didn't predict. In science, the most important results are often the ones that don't fit. Data that confirms a prediction is satisfying but rarely transformative. Data that contradicts a prediction forces the entire model to be reconsidered. And in aging research over the past decade, a handful of results have appeared that don't fit cleanly into any existing model of how aging works, or how reversal should behave. The brain reprogramming anomaly is one. When partial Yamanaka factor expression was applied to aging neural tissue, a small population of cells didn't respond the way the model predicted. They didn't rejuvenate cleanly. They didn't die. They entered a state that showed markers of both youthful gene expression and a different, previously uncatalogued pattern that the researchers couldn't assign to any known cell stage. These cells persisted in the tissue for weeks after the reprogramming factors were withdrawn. They didn't cause cancer. They didn't appear to harm the surrounding tissue, but they existed outside the established vocabulary of cell biology. The research teams involved have published their findings carefully, describing what was observed without overclaiming on interpretation. The scientific community's response has been genuine uncertainty, which is the appropriate response to a genuinely uncertain finding. A second unexpected result came from the young blood parabiosis experiments. At a more detailed level, when researchers analyzed which specific signals in young blood were driving the improvements in old tissue, they found that some of the benefit appeared to come not from adding positive factors, but from diluting negative ones. Old blood contains elevated concentrations of certain proteins that actively promote aging. When these were reduced simply by diluting the blood with a saline and albumin solution, some tissue improvements followed. A small clinical study called the Convoy Plasma Dilution Trial tested this in humans. Participants received a procedure that diluted their own blood plasma. Inflammation markers dropped. Some cognitive measures improved. The implication was disorienting. The problem wasn't just the absence of youth. The presence of age was actively causing damage, and removing it, even without adding anything young, produced benefit. This shifts the model. Aging isn't just the loss of a positive state, it's also the accumulation of an actively hostile one. And clearing the hostile state is itself a meaningful intervention. A third anomaly emerged from epigenetic clock research. When certain tissues from long-lived exceptional humans, people who lived past 110 years with good health, were analyzed for epigenetic age, the clocks in some tissues showed ages far lower than expected. But other tissues in the same individuals showed ages close to chronological. The protection wasn't uniform. Something in exceptional human aging allows certain tissues to maintain epigenetic youth while others age normally. What that something is remains unknown. Whether it is genetic, environmental, metabolic, or some combination is an active and unresolved question. But the finding suggests that the most successful natural human agers have found a way, through mechanisms not yet understood, to protect specific tissues from the epigenetic aging that affects everyone else. If that mechanism can be identified, it could represent the most powerful natural template for aging intervention that exists in human biology. In a handful of research centers around the world, scientists have been studying a specific group of people called Superagers. These are individuals over the age of 80 whose cognitive function, when measured rigorously, matches or exceeds that of healthy people 20 to 30 years younger. They remember conversations. They learn new skills. Their processing speed stays sharp. Their memory consolidation functions as it did in middle age. When their brains are scanned and compared to those of typical 80-year-olds, the differences are visible. The cortex, the outer layer of the brain responsible for higher cognition, is measurably thicker in superagers. Less of the grey matter has been lost to the decades of attrition that affects almost everyone else. What separates superagers from the rest is something researchers are working intensively to characterize. Genetics plays a role. Several gene variants have been identified that correlate with exceptional cognitive longevity, some relating to inflammation control, others relating to synaptic maintenance and neural connectivity. But genetics alone doesn't explain it. Some superagers don't carry the identified favorable variants. Some people who carry those variants don't show superager cognition. The genetic contribution is real and measurable, but not deterministic. Lifestyle factors show consistent associations. Physical exercise, especially cardiovascular exercise, sustained across decades, correlates strongly with preserved brain volume and cognitive function in aging. The mechanism connects directly to what we covered about mitochondria and neuroplasticity. Exercise drives mitophagy in brain cells, improves blood flow to neural tissue, and stimulates growth factors that support neuron maintenance. Social connection is a consistently replicated finding across superager populations. Individuals with robust, active social lives into old age show better cognitive preservation than isolated individuals with equivalent genetic profiles and physical health. The mechanism appears to involve the cognitive demands of social interaction, which continuously challenge and maintain the neural circuits responsible for memory, language, and emotional processing. The superager research matters for aging science because these individuals demonstrate, without any external intervention, that the biological deterioration we associate with very old age is not universal. It is common, but not inevitable. Some combination of genetic and environmental factors can hold the most important aging processes at bay in at least one organ, the brain, for an extraordinarily long time. This creates a research target of extraordinary specificity. If we could identify and replicate what superagers' brains are doing differently, we would have a biologically validated roadmap for cognitive longevity, drawn from actual human examples, not from mice, not from jellyfish, not from theory. The field is moving toward that roadmap. Longitudinal studies tracking superagers from middle age are underway, collecting genetic, metabolic, inflammatory, and lifestyle data at regular intervals. The dataset being built will eventually be large enough to separate the contributing factors with real statistical confidence. But while researchers chase the mechanisms behind exceptional human aging, the tools for intervening in ordinary human aging have been advancing on a separate, accelerating track. And those tools are converging on a moment that researchers describe carefully, almost reluctantly, as a potential clinical turning point. The progression from animal experiment to human trial is the most consequential step in any medical research program. It is also, in aging science, the most delayed one. The gap between compelling mouse results and human application in aging biology has historically stretched across decades, partly because of regulatory complexity, partly because of the challenge of measuring outcomes in organisms that live for 80 years rather than 2. The first generation of partial reprogramming human trials is now underway. Several biotechnology companies, including Altos Labs, New Limit, and Retro Biosciences, have initiated or are preparing early-stage human trials targeting specific tissues. The approach in early trials is deliberately narrow. Rather than attempting systemic whole-body reprogramming, which carries risks that aren't yet fully characterized. The initial trials target specific organs or conditions where the risk-benefit calculation is clearest. One approach targets the skin. Skin cells are accessible, non-vital in the way heart or brain cells are, and their aging is both measurable and visible. Early trials using topical or injectable delivery of reprogramming signals to skin tissue have been initiated with the primary goal of demonstrating safety. The question being asked is whether partial reprogramming can be applied to human cells in a living person without triggering uncontrolled responses. A more ambitious approach targets the immune system directly, attempting to use partial reprogramming to reset the epigenetic age of immune cells, restoring their function without altering their specificity. The theoretical benefit is significant: a younger functioning immune system, with the same learned immunity the individual has built across a lifetime. Epigenetic age measurements are being used as primary endpoints in several of these trials. The logic is that if the Horvath clock and its successors can reliably measure biological age, then moving the clock is a valid proxy for demonstrating that the intervention is doing what it's supposed to do. The regulatory path for these trials varies by country. In the United States, the Food and Drug Administration requires that trials target a specific disease or condition. Trials framed around aging as a general condition face categorization challenges. Some companies have navigated this by targeting specific aging-related conditions, treating the condition as the regulatory indication while measuring biological age as a secondary endpoint. The data from these early trials will take time to mature. Safety monitoring is extended. Follow-up periods are long. The results that will matter most, whether the interventions are safe, whether they produce durable epigenetic age reversal, and whether epigenetic age reversal translates into measurable health improvements, will not arrive quickly. But the trials exist. They are running. Human cells in living human bodies are being exposed to partial reprogramming signals for the first time in controlled medical settings. The results, whatever they show, will be the most consequential data the aging field has ever generated. And the question that no trial endpoint can fully answer is still waiting underneath all of it. The question of what reversal actually means when you apply it to a system as complex, as interconnected, and as deeply personal as a human life. There is a framework that helps clarify what partial reprogramming is actually doing, and why the results are simultaneously impressive and complicated. Think of your genome, the DNA sequence itself, as the hardware of a computer. The hardware doesn't change much across a lifetime. The sequence of base pairs you were born with is largely the sequence you carry at 80. Mutations aside, the hardware is stable. Your epigenome, the layer of chemical tags controlling which genes are expressed, is the software. It's what the hardware runs, and like software, it can be updated, corrupted, or partially reset without changing the underlying hardware at all. Aging, on this model, is partly a software corruption problem. The epigenetic tags that were correctly configured in youth, keeping the right genes active and the right genes silent, accumulate errors over time. Some genes that should stay quiet become active. Some genes that should stay active go silent. The cell's programming degrades even though its DNA hardware remains intact. Partial reprogramming is a software update. It resets some of the corrupted tags toward a younger configuration without touching the hardware. The DNA stays the same. The expression patterns shift. This is why the results in tissues like the retina are so clean. Retinal cells that received the reprogramming signal showed a shift in gene expression toward younger patterns, and function improved accordingly. The hardware was always capable of better performance. The software just needed to be corrected. The complication in the brain, as we've discussed, is that the software in neurons isn't just functional code. It is also the record of experience. The epigenetic configuration of a neuron at 60 is partly corruption and partly biography. The same molecular layer carries both. This framework also clarifies the danger of full reprogramming. Reformatting a hard drive removes corruption. It also removes everything else. Full Yamanaka factor induction takes the cellular software all the way back to factory settings. The cell becomes capable of becoming anything again, but everything it learned, everything it had become, is gone. The goal of aging reversal research is to find a way to remove the corruption without touching the biography, to update the software's performance without clearing the personal files. Whether that level of precision is achievable across the full complexity of the brain is the deepest unsolved problem in the field. And there is a further complication that this framework reveals. If the epigenome is software that runs on DNA hardware, what happens when the hardware itself has accumulated damage? Somatic mutations, DNA strand breaks, and copy number changes accumulate across a lifetime and represent actual hardware degradation. Resetting the software doesn't fix corrupted hardware. This means aging reversal probably cannot be accomplished by epigenetic reprogramming alone. A complete approach would also need to address the DNA damage underneath the epigenetic layer, the protein crosslinks in tissues, the senescent cell accumulation, the mitochondrial quality decline, and the immune system deterioration that allows all of these to build up unchecked. It is a stack of problems. Each layer is being addressed by different research groups using different tools on different timelines. The field has yet to run a comprehensive combined intervention in any organism that targets all layers simultaneously. When that experiment eventually runs, in mice first, and eventually in humans, the results will either confirm that the layered approach produces the compounded benefits the models predict, or reveal surprises that force the models to be rebuilt from the ground up. In the spring of 2018, a man in his early 60s flew to a clinic in Tijuana, Mexico, and paid a substantial amount of money to receive an infusion of young donor plasma. He had read about the parabiosis experiments. He had followed the early human trial results from plasma infusion studies. He decided not to wait. He was not alone. Across the United States and internationally, clinics offering young plasma infusions, unproven peptide cocktails, NAD+, drips, and combinations of compounds that had shown promise in animal studies began attracting clients willing to pay for access to experimental biology outside of formal trial settings. The Food and Drug Administration issued a warning in 2019, specifically targeting the commercial plasma infusion market, stating that young donor plasma had no proven clinical benefit and posed real risks including infection, transfusion reactions, and volume overload. The warning didn't stop the market. It simply confirmed that the market existed outside the regulatory framework designed to protect patients. This is what happens when science generates compelling signals faster than safety validation can follow. People who are aging, who are watching cognitive function decline, who have the resources to act, do not want to wait 15 years for randomized controlled trials to confirm what animal studies and small human pilots are already suggesting. They make individual risk calculations and pursue the most promising available options outside the system. The consequences are several and compound each other. People receive interventions of unknown safety and unverified efficacy, potentially harming themselves while generating no useful scientific data. The anecdotal reports they generate, positive or negative, contaminate the public understanding of what the science actually shows. Regulatory agencies, reacting to the uncontrolled market, may apply restrictions that slow legitimate trials. And the resources flowing into unregulated clinics could otherwise be funding the rigorous research that would answer the questions definitively. The regulatory system was not designed for this situation. The clinical trial framework assumes a stable boundary between the experimental and the approved, and that people will wait on the approved side while the experimental is evaluated. Aging has dissolved that boundary for a sufficiently motivated and resourced segment of the population. Some researchers argue that the solution is accelerated approval pathways for aging interventions, modeled on the mechanisms used for breakthrough cancer therapies, where promising early data can support provisional approval with ongoing monitoring. Others argue that the risks of undertested aging interventions, which could include cancer induction, immune disruption, or subtle neurological changes that take years to manifest, justify the slow, rigorous approach. The debate is real, the stakes are high on both sides, and no resolution is currently in sight. What is clear is that the people making decisions at the frontier of aging science, both in research labs and in unregulated clinics, are operating with incomplete information about a system of breathtaking complexity. And the most honest researchers in the field will tell you that the biggest surprises in this science are almost certainly still ahead. At this point in the story, it's worth being precise about where the science actually stands, because the distance between confirmed and theoretical in this field is enormous, and conflating them leads to serious misunderstanding. What is confirmed, reproducible, and not seriously disputed by mainstream biology? Telomere shortening drives cellular senescence. The Hayflick limit is real. Senescent cells actively damage surrounding tissue. Senolytics can selectively clear senescent cells in animals and show early promise in humans. The epigenetic clock measures biological age accurately and can be moved in humans. Partial reprogramming reverses epigenetic age in specific tissues of living animals. Vision was restored in aged mice using partial reprogramming. Thymic regeneration in adult humans is achievable with measurable immune improvement. NAD+ declines with age, and its precursors safely raise levels in humans. Young blood parabiosis improves organ function in old mice through mechanisms that include both adding positive factors and diluting negative ones. What is well supported, but still being characterized, whether epigenetic age reversal in humans translates into meaningful health span improvement. The specific molecular mechanisms responsible for the parabiosis effect, the long-term safety of NAD+ precursor supplementation at therapeutic doses, whether senolytic treatment in humans extends healthy lifespan as it does in mice. What is genuinely unknown, contested, or theoretical, whether a hard biological ceiling on human lifespan exists and where it sits, whether partial brain reprogramming can improve function without affecting memory or identity, whether combination therapies targeting multiple aging mechanisms simultaneously produce compounded benefits, whether aging reversal technologies will become broadly accessible or stratify along economic lines, what the unexpected non-classifiable cells observed in brain reprogramming experiments actually represent, and whether they pose any long-term risk. This map of certainty matters, because aging is a field where motivated reasoning runs high. People want aging to be solvable because they are aging. Researchers want their interventions to work because they have devoted careers to them. Investors want returns. Patients want treatments. The pressure to overpromise is enormous, and the cost of false hope is real. The actual science is extraordinary enough on its own. Confirmed results in this field, achieved within the past 20 years, have restructured what biology considers possible. The fact that partial reprogramming reverses aging in a living organ is remarkable. The fact that the epigenetic clock can be moved backward in humans is remarkable. The fact that thymic tissue can be regrown in adults is remarkable. Extraordinary confirmed results do not require embellishment. They require clear communication. And the clearest thing the science currently communicates is that aging is not a wall. It is a set of interacting processes, each of which is addressable, none of which has been fully addressed, and all of which are being worked on simultaneously by a global scientific community moving faster than at any previous point in history. The aging research field in 2025 looks nothing like it did in 2010. The capital flowing into it has grown by orders of magnitude. The tools available have advanced from broad genetic manipulations to precision molecular interventions. The measurement systems, led by epigenetic clocks and single-cell sequencing technology, can now characterize biological aging at a resolution that was impossible 15 years ago. What the next decade is building toward, based on the current trajectories of the research, falls into several converging directions. First-generation aging reversal therapies for specific conditions will likely reach regulatory approval in some jurisdictions within the next decade. These will be targeted interventions for specific age-related diseases, framed within existing regulatory categories, but measuring biological age as part of their outcome data. They will begin generating the human evidence base that the field currently lacks. Senolytic drugs will almost certainly reach broader clinical approval. The animal evidence is strong, the early human safety data is encouraging, and the mechanism is well enough understood that the risk profile is manageable. Clearing senescent cells from specific tissues in older patients with conditions driven by cellular aging is a near-term clinical target. Combination therapies will enter trials. Single-target interventions have consistently underperformed expectations in complex biological systems. The field is converging on the understanding that aging requires a combination approach, similar to how cancer treatment moved from single-drug chemotherapy to precisely tailored multi-agent regimens. Designing those combinations, testing them safely, and measuring their effects will occupy much of the next decade's clinical research. Epigenetic clock technology will mature into a clinical diagnostic tool. The ability to measure biological age with high accuracy across different tissues and cell types will transform how aging interventions are evaluated and how patients make decisions about their own health. Biological age testing will likely become routine in preventive medicine. The brain remains the hardest target and the most important one. The next decade's most consequential experiments will probably involve partial reprogramming of neural tissue in non-human primates, the step before human trials. Those results will either open the path to human brain aging reversal or reveal barriers that require fundamental rethinking. Either outcome advances the science. What the field cannot predict is its own surprises. Every decade of aging research has produced results that restructured the assumptions of the decade before. The discovery of telomerase, the Yamanaka factors, the epigenetic clock, the parabiosis effect, each arrived as a surprise that opened new directions no one had planned for. The next surprise is almost certainly already in the data somewhere, in an anomalous result that hasn't yet been recognized as foundational. Most of what we've covered in this exploration sits at the edge of medicine, in trials, in animal labs, in regulatory debates. But some of it is already happening, confirmed and available, in ways that most people haven't connected to the larger story of aging reversal. Exercise is the most powerful confirmed aging intervention available to anyone right now. This is not a soft lifestyle recommendation. The biology is mechanistic and precise. Regular cardiovascular exercise drives mitophagy, replacing damaged mitochondria with healthier ones. It stimulates the production of a protein called brain-derived neurotrophic factor, which supports neuron maintenance and connectivity. It reduces systemic inflammation, improves NAD+ metabolism, and maintains the cardiovascular health that keeps every organ supplied with oxygen and nutrients. Longitudinal studies tracking people across decades show that sustained exercise is the single strongest predictor of cognitive preservation into very old age. Sleep is the second underestimated aging intervention. During deep sleep, the brain's glymphatic system, a network of channels that flush metabolic waste from neural tissue, runs at full capacity. The amyloid proteins associated with Alzheimer's disease are cleared during sleep. Epigenetic repair processes that are suppressed during waking hours run during sleep. Chronic sleep deprivation measurably accelerates the epigenetic clock. Consistently getting adequate sleep is in biological terms a direct contribution to slowing biological aging. Diet's effect on aging runs through multiple pathways we've covered. High fiber intake supports microbiome diversity. Reduced processed sugar intake slows protein cross-linking through glycation. Caloric restriction, or its functional equivalent through intermittent fasting, activates many of the same pathways that NAD+ supports, including sirtuin activation and mitophagy. The Mediterranean diet pattern shows consistent associations with reduced biological age in large population studies, with inflammation markers and epigenetic clock readings both trending favorably. Social connection, which we mentioned in the context of superagers, operates through direct biological pathways. Chronic loneliness elevates cortisol and inflammatory markers, accelerating the same senescence cascade that drives physical aging. The effect is measurable and significant. Studies tracking isolated versus socially connected older adults show divergent biological aging trajectories, even when controlling for other health factors. These interventions don't require a clinical trial, a prescription, or a biotech startup. They work through mechanisms that are fully understood and confirmed at the molecular level. They are available to anyone, and they are probably responsible for a larger share of the variance in human aging than most people realize. Because the lifestyle factors that accelerate aging, chronic stress, poor sleep, physical inactivity, ultra-processed diets, and social isolation are also the factors most associated with poverty, shift work, and urban environments. The revolution in aging science is genuinely extraordinary, and the most important chapters of it may involve not just what the labs discover, but how broadly the confirmed tools already in hand are made accessible. There is one question underneath all of this that the science has not answered, cannot yet answer, and may take a century to resolve. It is not a technical question. It is a question about what we are actually trying to do. When we talk about reversing aging, we are talking about preserving the biological capacity to function, keeping the cells healthy, the brain sharp, the body capable. That goal seems clear, but function in service of what? A longer, healthier life is only as meaningful as what fills it. The assumption embedded in aging research, usually left unspoken, is that extending health is self-evidently good, that more years of capacity equals more years of value. For some people, for most people, that's probably true. More time with people you love, more time to contribute to things that matter, more years to understand the world more deeply. But the history of medicine contains enough cautionary stories about life extension without quality to make the assumption worth examining. Early dialysis extended lives that were sometimes spent in suffering. Early ventilators maintained biological function in patients whose quality of experience was negligible. The lesson that medicine took from those cases, after significant moral wrestling, was that biological life is not the terminal value. The capacity to experience, to connect, to pursue meaning is closer to the terminal value, and biological life is the substrate that makes it possible. Aging reversal, if it succeeds, extends the substrate. It does not guarantee that the experience filling the extended time is meaningful. That is a problem biology cannot solve. It is a problem each person has to solve for themselves across the full expanse of whatever time they are given. This is not a reason to slow the science. Chronic disease, cognitive decline, and physical deterioration are forms of suffering. Reducing them is straightforwardly good. But it is a reason to approach the longer life question with more intellectual honesty than the field sometimes applies to it. The researchers building aging interventions are solving a biological problem with enormous technical sophistication. The question of what humanity will do with the solutions they build is a different kind of problem, one that requires history, philosophy, ethics, economics, and political science working alongside the biology rather than waiting for the biology to deliver its results first. That integration hasn't happened yet. The biological work is decades ahead of the societal thinking required to absorb its consequences. Closing that gap is probably the most important non-technical challenge of the next 50 years. We started with blind mice that learned to see again, with cells that forgot how old they were and began behaving as if time had run backward, with an enzyme that rebuilds what aging destroys, and 4 genes that can reset a cell's entire identity. We followed that thread through epigenetic clocks that measure your true biological age, through zombie cells poisoning your tissues, through young blood and naked mole rats and jellyfish that cheat death by becoming young again, through Thymus regeneration in living adults, through NAD+ and mitochondria and the gut microbiome's quiet influence over every other system, through the ethics of identity in a reset brain, through the economics of unequal immortality, and through the physics of entropy that sits under all of it, asking whether this project has a ceiling. What the science shows, taken honestly and as a whole, is this. Aging is a process, not a verdict. It is a set of interacting biological mechanisms, each of which is now partially understood, each of which is the target of active research, and some of which have already yielded confirmed interventions that work in living animals, and, in early stages, in living humans. The reversal is real, in the tissues where it's been tested. The consequences are vast, in the dimensions that most people haven't started thinking about. The surprises are ongoing, in the anomalous results that keep appearing at the edges of what the models predicted. The field is not promising immortality. The serious scientists in it are careful about what they claim. They are promising something more modest, but more immediately significant. That the compression of the healthy years into the final decades of life, the decade of decline that currently awaits most people between vigor and death, may be shortened, or delayed, or in some cases meaningfully reversed. A world where people remain cognitively sharp and physically functional into their 90s and beyond is not a science fiction premise anymore. It is a clinical research goal with active trials, measurable endpoints, and early confirming results. The experiment has begun. The data is coming in. And the most important finding so far is that biology, the system we thought was immutably designed to wind down, turns out to have kept its own repair manuals the whole time. We just had to learn how to read them.